What if a single genetic quirk from one person 7,000 years ago is influencing your health right at this very moment, perhaps even offering you a hidden superpower against one of humanity's most notorious viruses? You've probably heard of HIV, the virus that caused a global pandemic, but did you know that some people are naturally, almost completely resistant to it? For decades, the origin of this resistance was a puzzle, with theories pointing to Vikings or medieval plagues. Well, fresh research, literally just published, has thrown all that out the window, tracing this incredible trait back to a single individual who lived thousands of years before any Viking longship ever set sail. This isn't just some minor genetic tweak we're talking about. It's a mutation called CCR5 delta 32, a 32 base pair deletion in the CCR5 gene. Think of CCR5 as a doorway that HIV uses to get into your immune cells. This mutation basically locks that door. And now a groundbreaking study in the journal Cell, published on May 9, 2025, has pinpointed its origin. Scientists analyzed over 2,000 modern genomes and DNA from more than 900 ancient human skeletons, some dating back to the early Stone Age. What they found is stunning. This mutation wasn't around in the earliest samples. Then it suddenly appears and spreads like wildfire in the late Neolithic period, roughly 6,700 to 9,000 years ago. And get this, every single person today who carries this protective mutation, and that's about 10 to 25% of people with Northern European ancestry, inherited it from one single ancestor. One person living near the Black Sea in the Western Eurasian steppe between 7 and 9,000 years ago. That's the new discovery, and it changes everything we thought we knew about where this lifesaver came from. So why is knowing the ancient birthday of this mutation such a big deal? Well, CCR5 delta 32 isn't just a scientific curiosity, it's a cornerstone of HIV research. The very first person cured of HIV, the famous Berlin patient, Timothy Ray Brown, received a bone marrow transplant from a donor who had 2 copies of this mutation. His immune system was essentially rebooted with HIV-resistant cells. This led to drugs that block the CCR5 receptor, mimicking the mutation's effect. It's a genuine lifesaver in the modern world, but HIV only emerged in the 20th century. So the big question has always been, if this mutation is so good against HIV, why was it already so common in Europeans long before HIV existed? What ancient force made it spread? Before this new study, the leading theories were frankly all over the place. Some said the Black Death in the 14th century favored people with the mutation. Others pointed to smallpox, a virus that also uses the CCR5 receptor. There was even that popular Viking hypothesis, the idea that fierce Scandinavian warriors spread it during their raids and travels. Estimates for its age swung wildly from just 700 years ago to over 5,000. But this new paper, using advanced AI to analyze fragmented ancient DNA, has definitively shown those ideas are off by millennia. The mutation is far older than the Black Death, older than widespread smallpox, and certainly older than the Vikings. Its rise coincides with something much more fundamental, the Neolithic Revolution, when humans shifted from nomadic hunting and gathering to settled farming. This suggests the original selective pressure wasn't a medieval plague, but perhaps a whole host of new diseases that emerged as people started living in larger, denser communities, often with domesticated animals. The researchers found that this genetic lineage was strongly favored by natural selection between about 7,000 and 2,000 years ago. So what was really going on back then that made this mutation so advantageous? This new research doesn't just give us a new origin story, it corrects some major misunderstandings about CCR5 delta 32. First, its age and origin. Forget the Middle Ages or Viking longboats. We're talking Stone Age, a single ancestor on the Eurasian steppe. This wasn't something that popped up in multiple places. It's one ancient gift passed down through countless generations. Second, the selective pressure, it wasn't the Black Death. In fact, recent studies on Black Death victims show other immune genes, like ERAP2 were under selection then, but CCR5 delta 32 didn't show a significant jump. The real driver seems to be tied to the challenges of early agricultural societies. As humans settled down, new pathogens likely emerge. What if this mutation offered a broad advantage? Third, the function and benefit. This is where it gets really interesting. Losing the function of an immune gene sounds bad, right? But CCR5 is involved in inflammation. The researchers suggest that losing its function might have dampened certain immune responses. An overly aggressive immune system can be deadly. Think of cytokine storms in severe flu or COVID. So, in an environment teeming with new germs, a more tempered immune response, less self-damage, could have been a lifesaver. It's not about a stronger immune system, but a smarter, more balanced one. Less is more in this evolutionary tale. And fourth, the scope of its effects. This mutation doesn't just affect HIV. The study found it's part of a larger genetic package, a cluster of 84 linked variants that influences a whole range of health conditions, autoimmune diseases, cardiovascular issues, neurodegenerative disorders, some cancers, and even how people fared with COVID-19. So it's not a simple on-off switch for HIV. It's a complex player in our immune system with a wide range of potential upsides and downsides. This is crucial because it tells us that evolution rarely gives a free lunch. The implications of this are pretty mind-bending. First, there's the sheer coincidence. A mutation selected for thousands of years ago, for reasons we're still figuring out, just happens to protect against a major virus that only appeared in the last century. As the lead author put it, it's almost coincidental and very fascinating. Evolution works in mysterious ways, preparing our genomes for past battles, and sometimes those old shields just happen to work against new enemies. Then there's that counterintuitive idea that a less reactive immune system could be better. It makes you wonder, what exact ancient diseases did our ancestors face that favored this dialed-down immunity? Was it one big killer, or a constant barrage of smaller threats in those new crowded settlements? Future research might even find traces of these ancient pathogens in skeletal remains, connecting them directly to the spread of CCR5 delta 32. And why this one mutation? Are there other ancient genetic superheroes hiding in our DNA, waiting for their moment? We know about some, like the sickle cell trait protecting against malaria. But CCR5 delta 32 is unique for its strong effect and how common it became in one large population group. Why didn't it spread anciently outside Western Eurasia? Perhaps the diseases or migration patterns were different elsewhere. The medical implications are also huge. Knowing that CCR5 delta 32 is part of this bigger genetic network means we have to be super careful with therapies that try to mimic it, like gene editing. You might remember the controversy in 2018 when a scientist in China edited human embryos to disrupt CCR5, supposedly to make them HIV resistant. This was widely condemned, partly because we don't fully understand all the consequences of messing with this gene. This new study reinforces those concerns. You can't just flip a switch without understanding the whole circuit board. For instance, while it protects against HIV, people with 2 copies of CCR5 delta 32 are more susceptible to severe West Nile virus. So it's a trade-off. But understanding this complexity could also lead to smarter therapies, maybe not by knocking out the gene entirely, but by gently nudging its activity. This also makes us think about all the other genetic mysteries we carry. What other ancient adaptations are lying dormant, ready to help or hinder us against future challenges? Evolution is all about these trade-offs. A gene variant that was a hero in one era could be a vulnerability in another. Take that ERAP2 gene variant that helped people survive the Black Death. Today, it's linked to an increased risk of Crohn's disease. It's a constant balancing act. The story of CCR5 delta 32 is sprinkled with some great tales. For years, that Viking blood theory was really popular. It painted a picture of Nordic warriors carrying this genetic shield across Europe. It's a cool story, but the science now points to earlier migrations, probably Stone Age steppe herders or Bronze Age traders, as the real distributors. It shows how science self-corrects and how our understanding evolves. Then there's its modern discovery in the 1990s. As the AIDS crisis raged, scientists found that some highly exposed people, like certain sex workers, just weren't getting infected. Many of them carried CCR5 delta 32. It was a massive breakthrough, showing a single gene could offer such profound protection. It was like finding an ancient key to a modern lock. And of course, the story of Timothy Ray Brown, the Berlin patient. His cure from HIV, thanks to a bone marrow transplant from a CCR5 delta 32 homozygous donor, was proof that this ancient immunity could be harnessed. It was a beacon of hope, a direct line from a 7,000-year-old genetic event to a 21st century medical miracle. But the flip side is the CRISPR babies controversy. The attempt to edit CCR5 in human embryos highlighted the ethical dangers and the scientific unknown. Knocking out a gene whose full effects we don't grasp could have serious, unintended consequences, like increased vulnerability to other viruses. This new study, showing the mutation is tied into a whole network of genetic effects, really underscores why that was so risky. So this single mutation, born in one individual on the Eurasian steppes 7,000 years ago, has had an incredible journey. It spread through ancient populations, likely helping them survive the new disease landscapes of early civilization. It lay relatively quiet for millennia. Then, bam, it becomes a critical factor in surviving a 20th century pandemic, HIV. It's inspired life-saving treatments, but also sparked ethical debates about gene editing our future. This latest research, pinning down its true age and single origin, adds a profound new chapter. It reminds us that deep within our DNA, we carry the echoes of ancient lives, ancient struggles, and astonishing resilience. What other ancient secrets are locked in our genes, waiting for science to uncover them, potentially holding keys to diseases we haven't even encountered yet? The story of CCR5 delta 32 shows our genetic past is always present. What will we discover next week? Make sure you're subscribed so you don't miss out on the latest deep research.